Fantastic watching Mike. Oh, that's the mouse. I'm going to cut half my jokes out now that it's recorded. Yeah, so thanks, Mike. It's been awesome watching Mike grow from a graduate student to an independent faculty member. I knew he was going to be successful. I bring up to our vice dean multiple times how we let him get away and we'll never get him back to UK now, that he's up here and loves Detroit and Wayne State and the support that he has up here. So I have not given a 45 to 50-minute seminar since before COVID, so this could go very poorly. So I apologize for that. Hopefully I live up to all of y'all expectations and you at least learn a little bit. I am a scientist, but I do enjoy career and mentor training and those types of things much more than the actual science. So I've really tried to incorporate some of those things into my talk today to make sure that you appreciate those things as well. So I talked to some of the trainees at lunch and I did my PhD in protein biochemistry and I hated it. Like I literally almost drove me out of science. My PhD mentor loves to hear me say that. But he saw the potential in me. So he just recommended that I go do a postdoc in something entirely different. So I went on to NIA and did that postdoc on caloric restriction and aging. And it always really instilled in me that passion for looking at, none of us really want to age, right? None of us really want to die. So how can we find ways to really delay the onset of those things, right? And this is a real person who turned 100, and there are exceptions to every rule, right? But generally what you want to do, right, is eat this healthy diet and be physically active. And really those are going to be the things that are going to increase your health span and your lifespan, hopefully, for the vast majority of us. But the problem is that these are some of my favorite foods. I went to school in Cincinnati, love Cincinnati style chili, lived by Philly, love a Philadelphia cheesesteak. And the problem is that many of us are eating proportions of high saturated fats and, are low in fruits and vegetables and that kind of thing, right? So this really got me thinking about, as I was doing these aging interventions across my whole life, I love to exercise, I love to eat healthy, but I don't want to have to do that for 80 or 90 years, right? So it got us into thinking about shorter term windows that we could do interventions, right? And on the left, I do a lot of work in mice and rats, and pregnancy is about 3 weeks and lifespan's about 150 weeks. And then in humans, my wife says it's 40 long weeks of pregnancy, but lifespan is about 4,000 weeks, right? So you're really talking about a 1 or 2% investment of time if you can streamline diet and behaviors during pregnancy to potentially influence the lifespan or health of your next generation. And these ideas aren't new, right? We've known for decades and even longer that drug and alcohol abuse or substance abuse during pregnancy can really lead to either birth defects or loss of pregnancy and a lot of these harmful effects that, again, we've known about for a long time. But really the whole DOHAD field, the developmental origins of health and disease, started to look at more of the subtle influences, right? So David Barker gets a ton of credit for the explosion of this field. And you can see I haven't given a talk since before 2020. So I stopped my PubMed updates in 2020. And then all of my software like SigmaPlot and those things are all expired. So I couldn't update the figure. But trust me, the explosion of PubMed citations in this field is really increasing dramatically over the decades. And David Barker found that, low birth weight individuals were at more risk for obesity and diabetes and cardiovascular diseases and other events later in life. And people thought he was crazy when he first published this because birth weight couldn't obviously impact later life diseases, right? But then through his collection of data and many others, then it started to become much more accepted. So here's you can't see at the bottom, but this was published in 2008 by Samuelson. I was not involved in this study, but they did a pretty cool experiment where they fed mice either a high fat or a normal diet. Those female mice gained weight, right? You're feeding them a high-fat diet, they become overweight. And then when they're pregnant, they're still way more. That's not terribly surprising. If you eat a high-fat diet, you become obese. What was more surprising is that the offspring that were born to these moms that were fed high-fat diets also ended up gaining more weight. You can see the body weight on the left there, or fat pad mass on the right. So both male and female offspring, if the mom was overweight, they had more fat mass, right? They've also seen differences in probably in a lot of different diseases, whether that's increased fat mass, hypertension, or impaired glucose tolerance. Again, not necessarily surprising though, because you've got this overweight female mouse that's where the developing embryo and fetus is coming from. So not necessarily as surprising, but there's also then, since then, has been people studying the impact of paternal influence, right? So feeding a high-fat diet to male rats led to female offspring that had impaired glucose tolerance, right? So this was published in 2010. I think, Mike, this is covering up some of my citations, but that's okay. If anybody wants these slides, you reach out to me, I'll make sure you get them. But anyway, so this is a little bit more surprising, right? That the paternal side of things was also having this influence. So certainly from the first time I gave this talk in 2008 or 2009, I've changed the way I look at these things to where we now have to think about paternal influence as well, right? And we're not just blaming the mom or blaming the dad. These are exposures that we're looking at. So right when I started my lab in 2009, this cover was on Time Magazine, how the first 9 months shapes the rest of your life, right? So really thinking, and this article is fantastic, if anybody's looking for a good quick summary from 15 years ago now, but it's a great article that kind of summarized and really kind of set me to where I was like, okay, well, maybe I am really looking in the right field. This is really awesome. And then one of my students found this in a magazine, and it was the bad daddy factor, right? So, but thinking about how we know all of these exposures in males can also have impact on offspring. So my whole lab overview is really summarized by this developmental origins of health and disease. And we were looking at exercise. So looking at a positive intervention, that's where we really started. We wanted to look at ways that we could improve health in the offspring. And we looked at a couple different things: diabetes, obesity, cancer. But then throughout the years that I've developed my lab, we also started looking at toxic exposures as well. So exercise during pregnancy, this is probably 2007 when I started doing these experiments. We knew that some women were exercising during pregnancy, but the general recommendation for pregnant women was to continue to do what you had been doing or even decrease your physical activity, right? Because we don't want to put that baby at stress or increase the risk of any kind of negative complications. There were many known benefits of exercise at the time, which is improved cardiovascular health, improved body composition and weight maintenance, less muscle discomfort and cramping, improved glucose homeostasis, and less labor complications. But no one really knew what was happening in the offspring if the mom exercised. There was some evidence in humans that maternal exercise would improve learning and memory, but there were unclear metabolic effects. So what we did was set out to really start to do some of these early experiments. And I kind of used some of the experiments and some of the protocols that we had from our aging experiments in mice and kind of did the same thing, which was we hypothesized voluntary exercise during pregnancy and nursing would improve health in the offspring. Very bland, not very detailed hypothesis. But we used these mice called the ICR or CD1 mice, which are an outbred stock. They're excellent breeders. They provide great maternal care. Gestation's about 20 days, and they have a litter size of 11 or 12. So it allows us to call back the litters to control for postnatal food availability and do some extra things that maybe we couldn't in other strains of mice. When we do the voluntary exercise during pregnancy, we had no negative effects in these offspring, right? We give them the running wheels prior to pregnancy, let the moms run as much or as little as they want, and then we track the health outcomes in the offspring. We don't have any differences in like pregnancy rates or number of pups per litter or pup weights or anything along those lines. The first experiments we were looking at, again, we were just looking at short-term intervention of maternal exercise, long-term health benefits in offspring. And we were looking first at obesity and diabetes. I came from an aging lab, so it was nothing to just to keep mice for years. So we started looking at this glucose homeostasis in the offspring. And once these mice reach adulthood, we see improvements in glucose homeostasis if those mice were born to exercise moms. So this is a glucose tolerance. I'll show a bunch of these curves today. You basically fast mice for a given period of time, give them either an injection or a bolus of glucose orally, and then you track blood glucose over time. In this case, you get the spike in blood glucose in all the mice, but if the mice were born to exercising moms, they had lower glucose levels, right? Which suggests that they're handling the glucose better. That can be summarized then by area under the curve. And you might look and you're like, oh, that's not that big of a deal. You're getting a 10 or 15% improvement in glucose homeostasis, but in reality, we're actually taking mice that were normal, right? Treated normal diets, everything was like perfectly what we would assume would be their best case scenario health-wise, but we can improve that, right? So for me, pretty exciting stuff. We also were looking at cancer and stress protection, which we haven't done much of over the last 5 to 10 years, but we also looked in a cancer model where we were doing skin painting with DMBA and TPA, which is the two-stage carcinogenesis model. And we did this in the offspring that were born to moms that were exercising or not. And when we treat them with these chemical carcinogens, we can go back in and count the number of tumors per mouse. And what we saw was a delay in the onset of tumors in those mice that were born to the exercising moms. And not only that, but they also have a decrease in tumor number. So for us, this was pretty exciting. We were able to get like a little baby RO3 grant for $50,000 or whatever, but we've never been able to turn this part of the project into like much larger funding. So we kind of let that project aside, but I think it's still pretty exciting that you look at how can these mice that are born to a mom that exercise be more protected against stress? So, but one of the things we kept asking ourselves is like, is this just a phenotype that we're seeing in one strain of mice and it's really not that important? So we basically reproduced exactly what we had done in mice and we did it in rats. And the mice, or the rats, like the mice, they run a ton. So this is kilometers per day. And this is how much the mice or the rats are running. You see they start about 1 kilometer per day, but they go up to about 7 kilometers a day. And then they do decrease their running after the pups are born. But we saw basically the exact same thing. In this experiment, we did slightly more intense experimentation where we did hyperinsulinemic euglycemic clamp, which is the gold standard for insulin sensitivity. And we saw that those offspring that were born to exercise moms were more insulin sensitive. So we were like, okay, this is great. This isn't just one model that we're seeing. So we were really excited about that. So we published this in about 2012, I think, in a couple different journals. But then what happened is all these other really awesome labs that were doing so much more than us were doing the same things that we were doing and potentially even better, right? So, which is great to see when other people see the same things that you do. But a lot of the individuals were adding high-fat diets and looking at obesity and the combinations of those types of things, which is pretty great. Again, there's lots of, and this hasn't been updated in quite a while, but there's a ton of data out there now looking at exercise during pregnancy and how it affects health in the offspring. But getting back to the point is like, do I really care if we can improve the health of a mouse, right? Like I want to improve the health of humans, right? Because that's the people we really care about. So we were like, okay, what can we do? I'm at University of Kentucky. We don't have a huge, like I hear about some of the clinical cohorts that are being set up here and expanded, and it's amazing, like makes me, I'll have trouble sleeping tonight because of like the amount of amazing things that y'all are doing up here and what we were trying to do at Kentucky. But I met with our CCTS director, our clinical translational director, and he's like, well, what can you get from a baby? I'm like, well, everybody's using placenta or cord blood. You know, this is the type of thing that I'm not terribly interested in just trying to replicate what other people are doing. We want to do something new, right? So he's like, well, the one thing you can get from a baby that would be of a substantial amount is the foreskin, right? And he was joking. And I was like, okay, well, I'm going to do that. And he's like, I don't think you should. So we set out, and for a while they called me one person at the NIH called me the foreskin guy. And I was like, that's probably not going to go over well for either of us. So, but we collected the foreskin after birth from about 50 babies. You know, we'd never tried to convince anyone to circumcise their child. We only interacted with those individuals that were already going to circumcise their child. But I was like, okay, this would be really cool because we can get about 1 gram of tissue. We can do RNA extractions. We can do protein. We can actually grow primary fibroblasts from the foreskin. I was like, this is beautiful, right? So we're going to be able to do this. And what we're going to do is just do like a pilot experiment. We're going to take all comers. We got 50 or so foreskins, and we're going to look at doing a questionnaire and say, okay, were you physically active during pregnancy or not, right? And then what happened is that we actually collected the data, and 3 out of the 47 women actually exercise during pregnancy, right? So unfortunately in Kentucky, there's not a lot of exercise happening during pregnancy. But what did happen is that we saw that, I found the creepiest photo I could online, but we found that like 13 or 14 of these women were smoking cigarettes during pregnancy, right? So what we then did was looked at some gene expression stuff in these foreskins from the babies that were born to smokers or not. And what we see is decreased birth weight, which we expected but the gestational length was the same because we only took in full-term babies, right? We were getting rid of those folks that were coming out preterm, but we still saw all these genetic markers that were like predisposing these babies to obesity and diabetes. So this led me into like a rabbit hole, like where I could talk a whole seminar about smoking during pregnancy. I can talk a whole seminar about nicotine and e-cigarette exposure during pregnancy, but I'm going to keep it on the environmental contaminant side moving forward. But the foreskin is a great model. We just never really got successfully funded because of the male-female sex differences and that kind of thing. So I'm not sure I would pick up a foreskin as a new junior faculty right now, but I think it was a great model for us to look at some of the things that we were interested in. But this leads back to toxic exposures, right? So we kind of accidentally led through this smoking exposure, but then around the same time, Dr. Bernie Hennig there in the middle was the director of our Superfund for, I don't know, 20 years or so. And this is a picture of when UK hosted the annual Superfund meeting in Lexington, Kentucky. And for those of you that don't know, this is Dr. Bill Sook, who recently retired as the director of the Superfund program. That's Dr. Linda Birnbaum, who was the director of the NIEHS. And this is our president at the University of Kentucky, Dr. Eli Capilouto. And everyone is laughing because Bernie introduced him as a postdoc instead of the president. So everyone's like dying laughing. But Bernie approached me and was like, hey, I want you to be a project on the Superfund. And I was like, I don't even know what that is. I don't know what you want me to do. And he's like, well, all you have to do is basically do what you're doing, but use PCBs. And I'm like, I don't even know what PCBs are. Well, I'm sure most of you do since you're environmental health related experts, but PCBs are these chemicals, right? There's 209 different ones where the chlorines could be substituted on the phenyl ring in different ways. Unfortunately, or fortunately for us, they were banned in 1977, but as a scientist, something that was banned in 1977 isn't probably a great project to pick up, but we did, right? And they are persistent organic pollutants because they last in the environment for a really long time. They're fat soluble, they're prone to accumulation, they accumulate in the food chain, so you're getting them through a number of different sources, either through food or through the air. They have a number of known health complications, including reproductive failure, fertility, miscarriage, and low birth weight. So these were some of the things that Bernie started getting me excited about. He's like, you should really start looking at PCBs. They do cross the placenta, they enter into breast milk, right? So Bernie approached me in, I don't know, 2011 or 2012 about working on this project. And right around then, there was a paper that came out that said prenatal exposure to PCBs could lead or at least contribute to obesity development in children, right? So we had all of this information. I was like, okay, let's do this. So we went out and set up a couple different pilot experiments where we did different exposures to PCBs. The PCB that Bernie recommended that we used was PCB 126, which may not mean anything to you, but it's the most potent activator of the aryl hydrocarbon receptor out of the PCBs. So for us, we always looked at this as a model. I never wanted to replicate what would happen in a human if you were exposed to like dramatic amounts of PCB 126, right? We were really trying to create a mouse model that would allow us to test interventions and look at mechanisms, right? So we came up with this exposure paradigm of giving mice, and we use different doses in some of these studies, and you'll see that sometimes I report it, sometimes I don't, but we generally see a lot of the same phenotypes in that if you give mice PCBs prior to pregnancy, during pregnancy, and during the nursing period, we then wean them and look at the outcomes in the offspring. And we almost always see a decrease in lean mass in these offspring. Fat mass, it really depends on the exposure paradigm that we use. Sometimes we see an increase in fat mass and sometimes we don't. And in the male offspring, especially, we see an impairment in glucose disposal if these mice were exposed to PCBs during the pregnancy and perinatal period. And this was all done by Dr. Sitiwe Rashid, who got his PhD in my lab, went on and did a postdoc with Becky Simmons at Penn, and we recently recruited him back to UK as faculty, and he gives an excellent talk. So if you're looking for additional folks to invite here, Dr. Sitiwe Rashid would be a great one to bring up here. So he basically set up the model, then went on and did other things in life. But Holly Swanson, who is my co-project leader, came onto the project because I really had no idea what I was doing with toxicology. So I linked up with her, and we've had just an amazing relationship over the past decade plus. And our first grant was really looking at, what about the timing of the exposure? Is it during the in utero period or is it during the nursing period? When is this important? Then we wanted to look at mechanisms as to why this was occurring. And then we wanted to test, I have maternal exercise, but we actually tested multiple interventions to see if this would protect offspring from outcomes associated with PCB exposure during pregnancy. And most of this work was done by Dr. Brittany Rice, who recently graduated from my laboratory. And she is also an amazing individual. She's doing a postdoc, and she's doing a postdoc at UK, but she's looking for positions now, but she is just dynamic. She actually presented 2 of these projects, which I'm going to present very poorly, and she won like at the National Superfund meeting for her poster. So if you want to see someone present this data well, invite her. So what about the timing of exposure, right? Our exposure paradigm to this point was giving prior to pregnancy, and during nursing. So we were like, okay, which of these periods is the most important? Is it gestation only? Is it nursing only? Like, when are these things the most critical? So our hypothesis was that, PCBs are going to cross the placenta. They're going to directly or alter that fetal or developmental programming and lead to obesity and or diabetes in these offspring. But there's also the exposure during milk. So the question is which of these is more important, or are they both required for these negative effects? So and I love clip art. Like I totally put these in my grants. Like I'm like, hey, if I'm drinking a glass of wine, I want to look at clip art while I'm reviewing a grant. Keeps me happy. So I put clip art in my grants. This was actually submitted, but we set up a beautiful experiment. Like this thing was perfect, right? So we were either going to expose these moms to vehicle or to PCB 126. This would be for our in utero environment, right? And then I don't have it on here, but we were going to cross-foster everybody, right? You'd either be cross-fostered to your own, like a different mom that was exposed, or you would be cross-fostered to a mom that wasn't exposed. So we would have this beautiful experiment to where you either were exposed only during pregnancy or only during nursing or vice versa, right? Well, unfortunately, the vendor that supplied our ICR CD1 mice like stopped supplying them. So then we had to get them from a different vendor. So we set up this beautiful experiment. Like Aya Cook hated me, like trying to rationalize the numbers of mice that we needed to use, because in developmental programming, we often just take one offspring per litter for a given purpose, right? So we have lots of large litters. Well, unfortunately, we had like major death and destruction because the dose wasn't quite right for this strain of mice that we were ordering from a different vendor. So we pivoted, right? That's what scientists do. We have to make sure that we're always aware of what's happening. And some of the data I'll show you today, especially for any of the trainees out here, is negative, right? Like we did not get the result that we anticipated. I had a PhD student that she had 3 negative papers. None of her hypotheses, or my hypotheses, worked. She published all 3 of them in subpar journals, right? But we honestly and ethically reported our stuff. And she was faculty within like a year and a half. She just got tenure and promoted. So just make sure you're reporting what you find, design your experiments appropriately, and be able to publish them. So that's my little sideline diatribe for trainees out there. But, you know, we took these mice then and we're like, okay, what can we do with those, you know, that didn't have pups and whatever. So we did postnatal exposure only, right? So we gave postnatal day 3, postnatal day 10, and postnatal day 17. We either exposed the mom to vehicle or PCBs, and then followed out the offspring. And some of my slides are really busy, but I'll try to walk you through them in the easiest way. And mostly, if you want the Cliff's Notes version, which I love, is at the top, right? So PCB exposure during the nursing period does not affect dam or offspring body weight. I don't have the dam, yeah, I do have the dam body weight. No, I don't have the dam body weight on here, but you can trust me. And we don't see effects on dam body weight, but you don't see it in the offspring either, right? Our females that were exposed to vehicle during nursing versus the PCB are the same. We do see the males weigh more than the females, but again, no PCB effect in those offspring. And we see that at either 12 weeks on the right or 3 weeks on the left. And we do wean mice at 3 weeks of age, just to kind of remind you of that fact. So again, no effects on body weight caused by the PCBs. But what about body composition? This is where we see some different effects in that paradigm where we give the 3 exposures. So once prior to pregnancy, once during pregnancy, and once during the nursing period. Again, you can look at the treatment p-value. On the left is the 3-week, right? On the right is the 12-week. And our treatment is causing no effect in fat mass, lean mass at 3 weeks or at 12 weeks of age. So again, no effects on body composition. If we look at glucose tolerance, again, these slides are pretty busy. So here's the glucose tolerance lines where we, again, fast the mice, track it over time. We can summarize that as area under the curve, right? So if you look in the 3-week-old offspring, our females that were exposed to vehicle during the nursing period, that's one of the controls, right? But we see an impairment in glucose disposal if those mice were exposed to PCBs during the nursing period, and we see the same thing in the males. And you can see that shown here with the p-value of less than 0.001. However, as we age those mice out, so to 12 weeks of age, we lose that effect, right? So the long-term programming of that glucose intolerance isn't there anymore, right? So it's a transient effect where it goes away. So we think that direct exposure during the nursing period has this effect on glucose tolerance, but it does go away over time. So I would think that's not a long-term programming effect like we see with the in utero exposures. So to summarize that part, we unfortunately didn't have the perfect cross-fostering experiment like we had designed, but what we did find was that nursing-only exposure led to normal body weight, normal body composition, and only short-term impairments in glucose tolerance. So we wanted to look at mechanisms too, right? And with PCB 126, especially, it activates the aryl hydrocarbon receptor, right? You get phase 1 detoxification through the cytochrome P450 pathways. But then there's also phase 2 response, right, which is controlled a lot in part by NRF2, so NFE2 related factor 2, which is a transcription factor that binds to the antioxidant response element, causes a upregulation in a bunch of detoxification enzymes. And what we wanted to do is look at mice that had either 2 copies of NRF2, 1 copy of NRF2, or no copies of NRF2. But in our pilot experiments, what we found was that giving mice PCB126, it didn't matter if they had 1 copy or 2, the response was the same. So that allowed us to only look at hets versus knockouts, which was pretty cool because that allowed us to set up our experiment. Again, sort of busy, but I'll try to walk you through it. We were able to set up an experiment where we had het moms, right? So they had 1 copy of NRF2. We either gave them vehicle or PCB, or we could use knockout moms that had no viable copies of NRF2 that were given either vehicle or PCB. And if we bred those to males of the other genotype, right, we would end up with offspring that would allow us to test, for offspring genotype. But since we set the experiment up like this, we could also look at maternal genotype as well, right? So was the NRF2 important in the moms or in the offspring? And in this experiment, it was pretty cool too, because we cross-fostered everyone to normal moms that had 2 copies of functional NRF2, and they were never exposed to PCBs, right? So this limits our PCB exposure paradigm only to that just prior to pregnancy and during the pregnancy period. And then we tracked our normal endpoints of obesity and glucose tolerance. And again, these figures are pretty busy, but what I'll show you is we kept these mice till 4 months of age, and I'm showing you the data from that age, but this is the offspring on the left to the NRF2 het dams. We have vehicle exposed on the left, PCB exposed on the right, blue or male, pink or female. What we wanted to look for were differences. We expected our knockouts to either be protected or more susceptible. I wasn't exactly sure which, but we actually saw no effect of offspring genotype in any of these outcomes when we're talking about treatment with PCBs. So my student was extremely disappointed, and she's like, okay, well, NRF2 genotype in the offspring meant nothing, right? But what we started to see was the maternal genotype meant something, right? So if we look, the one you really want to look at is treatment. So in these offspring to the NRF2 het dams, we see a significant effect on body weight in the offspring born to those het dams, but we don't see that in those offspring born to the knockout dams. So you're like, oh, well, okay, this is protective. Well, unfortunately, body weight is not just the be all end all, right? We need to look at body composition as well. So if you look at lean mass, which is on the top, the graphs are all set up the same way. We see a significant reduction in body weight or in lean mass if the offspring were exposed to PCBs, right? And we've seen that before, and you see that in both those offspring of the het dams, but also the offspring of the knockout dams on the right. If you look at the average fat, in the case on the left, those offspring of the het dams, we don't see any effect of the PCB treatment, but we see an increase in fat mass in those offspring that are born to those knockout dams. So again, the reason we didn't see the body weight in those offspring is because they had an increase in fat mass. When we look at glucose tolerance, again, pretty complex figures, but what we saw is that in those offspring from the het dams, we don't have a significant effect. There's a pretty strong trend towards an impairment in glucose tolerance, but that effect is much more significant in those offspring that were born to those knockout dams. So again, those knockout dams, for some reason, whether the PCBs are higher, we don't actually know the reason. We haven't done total PCB measurements or where those PCBs are going. But if those dams were knockouts, the offspring are much more harmed by those PCB exposures. So again, we published this, it's in 2021. So if you want more of the details, you can certainly find them there. Here's just a quick summary graph to where those offspring of the het dams had lower body weight, lower fat, lower lean mass, and slightly impaired glucose tolerance, whereas those from the knockout dams had higher fat mass, lower lean mass, and an impaired glucose tolerance. So again, some differences between maternal and offspring genotype in those studies. So again, I wanted to talk about interventions during pregnancy, and is this the right time to intervene? This is Sarah Tunlip, who's worked with me literally from the first month I was a faculty member, and she went and presented some of this work at the Superfund meeting this year. But I wanted to look at exercise, but Bernie Hennig is big into nutrition, and I had done some resveratrol stuff during my postdoc. So Bernie said, like, in order to be involved with the Superfund, he wanted me to do resveratrol. And there's a lot of evidence for why we would want to do that. I'll talk about that on the next slide, but we ended up setting up this experiment to look at whether or not resveratrol could protect against inflammation, glucose tolerance, oxidative stress, and improve the developmental programming of these offspring. So what is resveratrol? This is trans resveratrol. It's a polyphenol that's found in red wine, which is why people love to talk about it. It was really popular back in like the 2008 to 2015 range because it has a lot of beneficial health outcomes. Again, this is a graph showing the number of citations per year. So when it was discovered that it was present in red wine in 1992, there's a huge increase in the number of publications per year. And then Zhang and others published in 1997, it could protect against chemical carcinogenesis. So there was an explosion of papers that were published on resveratrol. And the beauty is that it opposes many of the negative effects of PCB126, right? It decreases oxidative stress and inflammation, whereas PCB126 increases this. It enhances insulin sensitivity, where we know PCB126 causes insulin resistance. And it's also been shown to be an aryl hydrocarbon receptor antagonist. So again, some pretty good evidence for why this thing might work. But why pregnancy? Resveratrol has also been shown to cross the placenta. It's been shown to protect against in utero ethanol toxicity. It's been shown to protect against TCDD or dioxin exposure, which also is the most potent activator of the aryl hydrocarbon receptor. And there was data that it protected against in utero hypoxia. So again, pretty exciting that there was some evidence that it would be protective. And there were also was a group out of Colorado that was looking at resveratrol during non-human primate pregnancy, right? And they saw some pretty interesting benefits on some outcomes, but also some concerning outcomes as well on islet cell function. But anyway, so at least there was some evidence that resveratrol might be protective and be worth looking at. I would not recommend drinking wine during pregnancy or taking resveratrol. I am just a PhD, not an MD though, so my recommendations probably don't matter that much. But so we set this experiment up in the exact same way that we had done all of our previous experiments, which is that we did 3 exposures to PCB126, 1 prior to pregnancy, 1 during pregnancy, and 1 during that nursing period. But what we did was we fed diets that either had resveratrol or not for 2 weeks prior to that start of the exposure paradigm. And then we measured those same outcomes in Offspring. And you can't see it here, but we used 0.1% resveratrol in the diet, which was similar to some of the experiments that we had done when I was a postdoc at NIA that showed a slew of health benefits. That dose of resveratrol is much higher than you could get from red wine. It would have to be like a supplement type dose, right? So much higher than what you would get from a normal dietary exposure. But what we saw, again, lots of the experiments in my lab turn out how we don't expect them to. What we saw is that you can just look at the resveratrol p-value. While PCBs had a big effect on body weight, resveratrol had no effect. That's in both the males and in the females. If we look at the effect on lean mass, again, PCBs are causing a reduction in lean mass, but resveratrol had no effect on resveratrol. If we look at area under the curve for glucose tolerance, the males, we have a significant effect of the PCBs, whereas resveratrol had no effect. And oftentimes in our experiments, I think because we're right on the edge of the effective dose during pregnancy, we don't always see the negative effects in the female offspring. And I mean, I've talked to some other folks here today and other individuals that see that the females aren't as susceptible to some of these environmental exposures. But again, kind of a sad day for the person that was working on the project, but I'm trying to get Bernie Hennig, who's stepping down as the editor of Journal of Nutritional Biochemistry in the coming months. I'm trying to still get him to publish this, since we only did the experiment because he thought it was a really good idea. So even though it didn't quite work out as expected, we are working to publish this. This is sitting in my inbox. And some words of caution is that we only chose 1 dose of PCB126 and 1 dose of resveratrol. It's not like this was a dose effect to where we really set out to see this. But again, we collected the data. I think it's only fair to make sure that we report it in the literature. But what I really cared about was the behavioral intervention of maternal exercise, right? That is what we had set out to begin to do anyway. So it was kind of like a nice na-na-na-na when I got to tell Bernie that the resveratrol didn't work, but we were then going to test maternal exercise, right? So in this case, we did use the lower dose of resveratrol because this was right around the time that strain switch had happened from the vendors. But in this case, we did the same maternal exercise intervention where they could voluntarily exercise as much or as little as they wanted, and we exposed them to PCBs prior to, during pregnancy, and then during the nursing period. And what we saw, and we're only showing the males here because the females we used for other experiments, and we also didn't see the glucose tolerance effects and things. But if we expose those mice to PCB 126 and they were born to sedentary vehicles, so our overall control group, they weighed this amount. But if we gave them PCBs, they actually had an increased body weight. And we think this is, again, because of the lower dose that we gave to them. But if the mom exercised and was exposed to PCBs, the offspring looked just like the overall control group. And that difference in body weight was made up by fat mass. When we did echo MRI and looked at that, it seems to be driven by a decrease in fat mass in those offspring born to the exercise moms. So pretty exciting. When we do glucose tolerance, again, these curves are pretty overlapping and things, so it's difficult to see. So we show that under area under the curve. And what we see is, again, there's a significant increase in the area under the curve, so worse glucose tolerance in those offspring that are born to PCB-exposed sedentary moms, but there's a pretty strong trend towards protection in those offspring if the mom exercised. So again, pretty exciting for us. We're working on this paper. We did repeated measures over time. I'm showing you one time point. It was consistent across all of those, but it's a statistical nightmare for a normal lab that does basic science research. So we're working with the statistician to make sure we accurately represent all these data. But again, pretty exciting because we're adding a toxic exposure with our exercise paradigm and seeing this protection. However, we did want to look to see, we didn't look at mechanism at all, but what we wanted to see is what was happening in some of these, the male and female mice. But we took the livers, right? So we don't keep all of the offspring for their entire life. We actually collect the livers and some different tissues from the mice during the nursing period. And what we did was looked at some of the markers of gene expression. So CYP1A1 is dramatically increased in PCB-exposed offspring. And what we saw was that exercise didn't have any effect on that, right? So we were still getting the same increases in CYP1A1, or NQO1 is downstream of NRF2, and we see significant increases in that. So at least it didn't seem to be driven by levels of these gene expression profiles in the liver. So we're not exactly sure why the improvement is happening. It's probably not at the surface of the liver anyway. It's probably in skeletal muscle or adipose tissue, but we've got a lot more work to do on this project if we want to look at mechanisms and other things. So just to summarize again, with sedentary PCB offspring, they're altered with body weight, body composition, glucose intolerance, and gene expression. And exercise in those PCB-exposed offspring improves all of those except the gene expression profile. It's the same whether the mom exercised or not. So just last week, you'll probably recognize numerous names on here, but we used the female offspring and collected the cecum from those mice and published a collaborative cross-center and cross-institution paper. And Mike, I'm sure, would love to tell you about this study at some point in the future. But anyway, pretty exciting. Just got this published a week or two ago. So congratulations, Manisha, first author on the paper. Okay, so our most recent renewal was looking at maternal exercise, but we also added an offspring exercise component to that experiment as well. So what if the moms were exposed to PCBs, and then could the offspring exercise rescue that phenotype? So we set out to do that experiment where we had 60 female mice that we either exposed to vehicle or PCB 126. We then bred them with males, and then we gave the offspring the ability to remain sedentary, gave them an opportunity to exercise with those running wheels, or we were, PCB sedentary or PCB exercise, right? So again, we use those running wheels and contract the data for the running in these mice. And we looked at first body weight. So offspring exercise lowers the body weight, but what you'll see is that, so the black triangles are the males that were vehicle exposed and were sedentary, whereas here's the vehicle exposed exercise. So they do have a reduction in body weight. But if you look at the PCB exposed offspring, they have an even further reduction in body weight, right? So they weigh less, and then exercising in that group causes an even further reduction. And we see that in both males on the left and females on the right. PCBs decrease lean mass, which we've seen before, right? So we've got male vehicle sedentary, male vehicle exercise, male PCB sedentary, male PCB exercise, right? So the PCB exposure in these mice causes a decrease in lean mass. And we see that in both males and females, obviously. And the problem is that the offspring exercise is having no further effect, right? So it's not protecting against that loss in lean mass in these offspring. So it's a bit unfortunate. And then we thought we would see this protection to where offspring exercise would end up improving these outcomes, and it didn't. Now, what the increase, or I guess the additional loss of body weight was caused by a reduction in fat mass in those offspring that were exercising, right? So in this case, we didn't see an effect on fatness in those PCB exposed mice, but we do in anything that's exercising, right? We see a reduction in that fat mass. So again, exercise is having benefits. It's just not having any benefits in response to that PCB exposure. If we look at area under the curve, again, so our glucose tolerance measurement, we see an improvement with exercise. So those lower area under the curves with exercise, but again, no interaction with the PCBs. So it's not really improving any of those PCB outcomes. So unfortunately, what did happen though is that the maternal PCB exposure does cause the offspring to run less, right? So if we give them open access to the running wheels, if they were exposed to PCBs in that perinatal period, they have a reduction in exercise, which is a bit unfortunate. And something we probably are going to look a little bit further at is whether that's they don't have the ability to exercise because they would get sore, or they don't have, they don't have the motivation to exercise, or what the reasoning is for that. But again, maybe we'll look at that, but probably not with us going in different directions with our Superfund renewal. But this was all presented, this is Nathan Boyce, who presented this just a couple weeks ago at NCUR, so the National Council for Undergraduate Research. He did a really awesome job. So he's working on the manuscript from this experiment, but again, didn't quite work out as well as we had hoped, where offspring exercise was unable to rescue some of those phenotypes. So in the kind of want to get the last couple slides, but we did another experiment where we dechlorinated PCBs. So DB Bhattacharya is our program, or our Superfund Project 3 PI, and he can dechlorinate PCBs by some magical method. I have no idea, I'm not an engineer, but he can get rid of these chlorines and make biphenyl very easily and cost effectively. So what he wanted to know is, was it non-toxic, basically, if we gave, in our paradigm, if we gave biphenyl instead of PCBs, right? So in this case, we actually set up that experiment. He did not dechlorinate them. We just bought 99.9% pure biphenyl, but we basically did the experiments the exact same way that we always do. This was done by two. Actually, she was a high school student that just got accepted into Emory and will be starting as a freshman there next fall. And a college student did these projects. So Hannah Greenwell and Nina Kerwell did this experiment together over the summer. And basically, I'm going to summarize this in about 30 seconds. PCBs caused all of the same things that we always see, and biphenyl is like perfectly harmless, which is pretty magnificent. In that, I guess as a toxicologist, you could look at biphenyl and think it's not going to be very toxic, but water in enough dose can be toxic. So anyway, the biphenyl seemed to have pretty much no effect compared to the control, whereas PCBs cause these negative effects that we typically see. So pretty exciting though, that we were able to get this. So we're working on publishing this as well. And if you look at gene expression again, CYP1A1, this is in the moms themselves, and then in the male offspring and the female offspring. Again, biphenyl pretty much caused no effects, which is pretty cool actually. And that's the summary there. So to close out our Superfund part, like we've trained a ton between Holly and me. We've had a blast training different graduate students, medical students, junior faculty. It's been awesome. Like I said, Brittany has won numerous poster competitions for a lot of the data that I presented because she presents it so much better. Josh Preston is an MD PhD student that's at Emory now, working with their P30 center down there. So it's been a lot of fun with cross collaborations in different centers, and we found a bunch of different things, which I already kind of summarized for you. But one of the things I want to just mention in the last couple of minutes is we had a third aim in that grant, which was we had a collaborator that had a beautiful cohort where she had collected data all across pregnancy and collected serum samples and then collected mesenchymal stem cells from the babies at birth. And we wrote up this amazing aim looking at glucose uptake, at adipogenesis, myogenesis, all in these mesenchymal stem cells. And we were going to measure PFAS and we were going to measure PCBs, and it was like reviewed beautifully. They loved it. But then NIH cut our budget by half, so we couldn't do the same. So I was like devastated because I'm like, this is the only aim I really was excited about. So again, you can only beat me down for so long. So we came out and I reached out to Eileen and I was like, hey, that sample we had, like, let's turn this into a whole grant, right? So we basically took that third aim, expanded it, made sure there wasn't overlap. And then we wrote a Victor application, which if you're not familiar with that, it's a virtual consortium where you cannot have published with the person before. So Eileen and I, within 6 weeks, grabbed Todd Everson from Emory, who was a postdoc to Carmen Marcet, who's one of my closest friends, and then Kartik Shankar. And within 6 weeks, we wrote our Victor application with literally no data. And we basically said, hey, we're going to do a bunch of really cool stuff in these cohorts that Eileen has, which are absolutely beautiful. That's us celebrating the grant that we got to be able to do this. So this is in Aspen, Colorado. The four of us are celebrating because we got the grant funded and we're collecting data. But like the cohort that I heard is going on here, we have very similar type things, and I've collected none of this. This is all Eileen and her team, but they've got about 300 women they bring in just prior to 10 weeks of pregnancy. They also have data in the fathers. Then they track these babies out till birth, follow them to 2 years, 5 years, 8 years. They collect mesenchymal stem cells, placenta at birth. They collect urine, serum, feces. They do all these body composition measurements. And her goal is obesity, diabetes, like a lot of the same stuff that I had, right? So she's got samples in each trimester from the mom's umbilical cord, like it's beautiful. So we're measuring the PFAS in these like right now. We have probably 40 samples to go in this first cohort. So I was hoping I delayed long enough that I could show data, but like it's just not quite there yet. But invite Eileen or Todd or Kartik and they can give you a talk. Lots of people have cohorts like that. This is the reason that I like started stalking Eileen and I was like, please work with me, please work with me. So she's got almost the exact same cohort, but they introduced a randomized control trial with exercise. So they brought these women in and had them start doing 3 days a week of exercise or provided standard of care, and then they collect all the same things, right? So she's got these beautiful cohorts and this amazing team of individuals that are working with her that collect these data, collect the time points, keep these folks coming back. And again, they're in Little Rock, Arkansas, so there's a pretty high level of PFAS down there. And then this is her funding comes from the USDA. That's how she's able to do a lot of this in her team. So I just wanted to make sure that I've done none of this. All I did was knew the 3 people involved and knew that I wanted to work with her. But anyway, our hypothesis for that is really looking at PFAS burden across pregnancy and then in the cord and looking at whether or not that affects postnatal birth growth, like the different things that she's already collecting. Todd and Kartik then are looking at placental PFAS concentrations and how that influences epigenetic regulation and gene expression. So they're doing all kinds of high-level data analyses and things with their aim. And then the aim that I'm associated with is the randomized control trial. And the question is will exercise affect any of those levels of PFAS across pregnancy or affect, birth weight and other things in that cohort? So again, we've actually taken the consortium very seriously. We meet every other week via Zoom. Wish we got to meet, in Atlanta or somewhere cool, but we meet via Zoom and actually talk, and we're working on 2 different grants right now because she has milk from all these babies too, and there's so little known about milk exposure and child growth and PFAS. So again, pretty exciting. Hopefully in a couple months, we'll be able to start sharing some of the data. So we're about to start analyzing the PFAS levels for AIM-3. So we don't have any of that done yet, but hopefully in the next couple of months, we'll start to get that data and see. The hope is that it causes no effects on, growth trajectories, but my guess is it will. And then Mike and I are talking about doing some exercise stuff with mice and looking at PFAS and metabolism and where it's going in response to exercise. Because even if, let's say, exercise causes a decrease in serum levels of PFAS, we're not actually sure whether that's beneficial, right? Is it causing it to go to the heart or the brain or to the baby? Those are a lot of things that we don't know. So we're just scratching the surface, I think, of some pretty exciting areas. And I do, in the last minute, sorry, Mike, I'm not leaving that much time for questions. This is Rachel Tinius, who I mentioned today. She's a faculty member at Western Kentucky, who I mentor. And if you can read her shirt, it says baby's first marathon. So she just ran Boston last week, 3.5 hours pregnant with her 4th baby. But she lives and breathes the exercise during pregnancy. She's an outstanding individual and someone that you should invite up here to give a talk as well. But I've been mentoring her, but really like she's basically dragging me along for the ride because she is awesome. But she created this app. So there's 40,000 fitness apps and 6,000 pregnancy apps, but none that overlapped for the 2. So she created this app, bumped up. I get no funds from this. She probably does. So if she came here, she would have a conflict of interest. I don't. But she created this app and with doctors and everybody on board to try to really increase physical activity and tracking of weight gain and things across pregnancy. And she's about to start a randomized control trial with her app to look at fitness and other things during pregnancy. So anyway, she's awesome. And these are the types of things that I think, whether we can start giving different nutraceuticals or pharmaceuticals during pregnancy and intervene that way, maybe not yet, but I think with exercise, it is something that's going to be safe and effective. So again, our long range goal is to continue to do these types of experiments and streamline diet and exercise during pregnancy. I have a ton of people that work for me and have worked for me and really drive the science. That was our lab right before COVID happened. It was awesome. Hopefully we can get back there someday and be large. And my daughter created this when she was like 9, and I try to show it all the time because it's kind of how we live our life. And, you know, leave people happier than you found them, or at least healthier than you found them? And that was my daughter then. And that's my last slide. And I will open it up to any questions. Yeah. Oh, thank you.
Did you look at that to some extent? And as an intervention, then why didn't you look at mycin as an alternative to that? Because that's next step, right?
Yeah, those are great questions, honestly. And we did not look at mTOR directly. I think a lot of our momentum fell off when we realized that the offspring weren't different depending on the NRF2 genotype. So I think we lost a little bit of momentum there, and my student wanted to graduate. So not great reasons for not looking at it, but yeah, that was kind of what we were thinking. But yeah, the rapamycin idea is a very interesting one. I don't know if you're pursuing that yet during pregnancy or at any points of intervention. Yeah. Okay. Right. Yeah. Yeah. So I mean, there's lots of exciting things to think about. I mean, I think those are things that if we had unlimited funds and unlimited people, we would probably do. But yeah, great question. Yeah. Yeah. Well, the resveratrol study started in probably 2012 or 2013. So that was when it was still fairly hot. But at this point, yeah, it's kind of fallen off a little bit in its likability. But I talked to some folks today about metformin was one that some other folks are looking at. Again, I think exercise is an easier one for me simply because it's unlikely to be harmful for the vast majority of pregnant women. Whereas if we start adding in, you know, pharmaceuticals or even nutraceuticals or other things, does make me a little bit nervous, especially with some of the branched chain amino acid data and things that are out there from the 80s and things. But yeah, great question. Yeah. Yeah. So the question is about whether or not, sorry, I should have done that first time for the Zoomers, but yeah, so the question is about adding baby teeth to the experiments. That's a great suggestion. We're starting to do that a lot at University of Kentucky. I'm not sure if Eileen at Arkansas is doing that. I need to talk to her about that because that is something that we could add very easily. Anything that falls out and like they don't need anymore, the tooth fairy, like my kids were never firm believers in the tooth fairy, but they are willing to donate their teeth to science for money. So yeah. Yeah, that's a great suggestion. Actually, I'll text Son as soon as I get a free moment today in my jam-packed schedule, I may send a text message to Arkansas. So thanks. Great. Anybody else? Oh God, Mike, do you want to pull those up while I answer this question?
Kevin, I'm going to guess you don't have a psychologist.
No. Did I say something wrong?
No, because if you had a psychologist in your team, they would insist that the third.
Okay.
Forced exercises.
Yeah. Yeah. So the question is about having a forced exercise group that would be basically doing the same thing as the voluntary exercise. We do not have that. We have, I didn't show it today, but we have tried to do, even though it's forced exercise, we have, picture a hot dog roller at 7-Eleven. You can put the wheels on top of that and you basically enclose the mouse and then they have to move. We've done that exposure paradigm with the controlled exercise. We call it controlled exercise, not forced exercise. And we don't see any benefits at all. And I think it's because it's adding a level of stress that if they're not wanting to do it, that they don't get that benefit. So I think while we didn't do the experiment that you're going to do, it's probably going to be very stressful. It's probably going to be very negative. And all of the things that we've done so far in our laboratory leads me to believe that any kind of stress during pregnancy is like bad, right? So that annoying, like poke my wife to try to get her to go do the exercise, like was that beneficial? Or if she would have just got up and like done that on her own, likely would have been much more beneficial.
But that's my point. You're taking a pregnant woman who possibly does not want to exercise, but you're going to tell her, I assume, in the future that she should exercise.
Yeah. Yeah, so I think it's all in the delivery. We talked about that at lunch, I think today with, well, and it's the non-blame game, right? Like
Yeah, but
it's the,
I'm saying that if you look at your effects, you find that the stress is having a pretty
Yeah.
result.
yeah, and I agree. And I think it's that matter of like, I think it's just changing the message and changing the educational platform of like making sure that instead of sitting like, okay, like don't just sit on the couch and throw your feet up and eat for two, right? Like that messaging should be long over, right? But that hasn't reached the entire like US, right? And I don't know. I mean, I don't think you need to go run a marathon when you're pregnant, or I don't, think you need to pick up a new like hobby or exercise when you're pregnant, but like getting up and walking around the neighborhood, I think is the messaging that I would like to make. Or like if you bring Rachel up here, I mean, she's fantastic. Like she complained to me or in an article, and then I asked her about it. But if you go to Bed Bath and Beyond or you go to Babies R Us and they have the mom parking, right, and it's like right in front, and she's like, these are the people that should be parking as far away as possible to get the extra steps, right? But it's that, how do you convey that without making it a blame game or like whatever? And I think I've at least tried to improve the way I give a talk, because we know there's paternal influence as well, right? We know it's a team effort and like that kind of stuff. And whether it translates through behavior or whether it translates through genetics, I mean, I don't know the answer to that yet. But yes.
One other question, interesting behavioral, whether the likes to exercise or transit. Curious if you've ever thought of doing something like on the front, like adding crowding, like in the cages, overcrowd the population density per cage, because that has inflammation exposure to the environmental type of effect that might both affect the behavior, but also the was there any like environmental type of thing like the front end while you're doing it? It's like that.
Yes. So the question is about adding extra stressors in crowd caging or cage crowding and others. And I guess we haven't thought about that specifically. I mean, I know in the aging community, like crowded litter will actually lead to, if you have less food availability early postnatally, that actually leads to a lot of health benefits, right? Because you get the early caloric restriction or lifespan part. But ACE, like the adverse childhood events, like lots of folks are looking at that in combination with environmental exposures to where, well, not lots of people I know are, but taking pups away from mom for a given period of time and adding that level of stress, and then adding a combined, environmental stressor as well. I mean, I think those things are, I guess the possibilities are endless on the amount of things that we can do. But yeah, I mean, I think those are important things that we need to start looking at, is like, how do we add all of these things together, and then how do we continue to improve them? But we haven't done that specifically, but yeah.
So do you think, I mean, it seems like imagine running a marathon to the baby during pregnancy. Do you think there's a limit how much exercise the mom should have? Maybe the mom should have
well, and I think the question is what was the person doing beforehand too, right? Like, so I think the big recommendation is not to change things too dramatically, right? Like, even if you're going to increase your exercise, like it shouldn't be going from never exercising to then doing something dramatic and something crazy. But for like Rachel, for example, and she'll tell you this, she ran 20 miles 5 times or 6 times in the last couple weeks before her marathon, right? Like, that's just her lifestyle, is she runs every single day and loves it. And she's got, it's something she did during all of her pregnancies and whatever. Now, if I went out and ran 26 miles today, I would die. I would literally be dead. So I don't know that the pregnancy or that part of it has as much to do with it just as physiological response and just that repetition and being able to build up to that, whatever it is. Again, I think running a marathon is crazy for any individual, because I don't understand as a human why a person would want to do that. But our motivations and our reward responses are all different, right? So for me, I capped out at 4 miles, but I'm also a 220-pound, like right, like the non-graceful runner. So, but yeah.
Women for girls who run marathons have to live puberty
and I guess it depends on if your endpoint aging in the aging community. I mean, delayed puberty is probably going to lead to longer life, right? So I think it depends on, again, the perspective of like, yeah, if your goal is to reproduce at 18 to 36 years of age, then maybe there would be a negative impact on that part. But if your goal is to lead live a long and healthy life, delayed puberty would actually probably be good. Yeah. I'm mid-40s now and I can't even imagine having a baby. I'm tired. Well, thank you all. If anybody wants to talk more, sorry, I went a little longer than I wanted, but I guess we're only at 1:33. So thank you.
Thanks, Kevin.